This lab is a three-parter, and this video covers just the first of the 3 weeks. Our focal topic, haha, today will be microscopes, and the basic objective, haha, is to get you all up to snuff when it comes to proper use of microscopes, as this is something that physiology and anatomy instructors really want to see in the students coming out of Bio 110 and into their classes. The first thing we'll address is how to handle the scopes and how they need to be put away after every lab. Our perspective here is that each microscope that we purchase, you purchase actually, as this is a tax-funded public institution, is just over $1,000. And we would like to keep them in use for 12 to 15 years before they're replaced with new scopes. In those 12 to 15 years, they're going to be used by hundreds of different students, and our ability to actually keep a scope in use for that time depends on our doing as much as we can to prevent mishandling of the scopes through those years. Now, from the student perspective, your skill in using the microscope will dictate your ability to see all the stuff that you're supposed to be seeing in this class, but also in your histology labs and anatomy, physiology labs, and especially in the microbiology labs, where you'll be behind a microscope for many hours each week. Moreover, you'll depend on your scope for access to important course content, and so knowing how to use your microscope and how to keep your microscope in top working order is just a great idea. Getting a microscope out of the cabinet and carrying it to your benchtop might seem like a pretty trivial thing. Sure, don't drop your scope, but there are a few really substantial points that need to be made to alert you to potential risks. First, the ocular lenses. They're not secured in the microscope headpiece, so tilting the scope, for example, as you're getting one off the top tier of cubby holes, can result in an ocular falling out. Second, there's this thin metal arm that's part of the stage control, the thingy that moves the slide around on the stage, that could be sticking way out from the side of the scope. That will be sticking out from the side of the scope when you're using it. Now this thing should be pushed in so it doesn't stick out before the scope is put away, but you can never be sure that the previous user did the right thing. And so you'll want to give a quick check to see that it's not sticking out from the side of the scope before you pull the scope out of the cubby. Third, the cord should be coiled and secured so that it doesn't dangle to the floor as you're getting the scope out of the cabinet and carrying it back to your space. And of course, use both hands to carry your scope, one hand holding the arm and the other supporting the base. When you get your scope back to your table, you can go ahead and unwind the cord, but before plugging it in, make sure that the light switch is off. The switch is a knob on the base of the microscope, and turning it all the way in one direction, it will click off. Having the light source off before plugging in will let the bulb and fuse last for the full 12 to 15 years of the scope's life. Plugging in with the light on high will put a surge through these delicate parts and dramatically reduce their lifespan. Binocular scopes have two eyepieces, and you'll want to take advantage of this feature. That is, rather than squinting one eye and using the other to look through the scope. Follow the instructions in the lab handout to determine your interpupillary distance and diopter settings. You'll do this while viewing the microscope slide with the printed letter E. The next thing you'll do is to set your microscope to ocular illumination. Again, follow the instructions in your lab handout. Some people say kolar illumination, but since the lab handout was printed using the umlaut on the O, we're going to pronounce that umlaut O in the correct way, and that's killer. Say killer, but stretch your lips out like you're doing a duck-faced selfie. Ocular illumination is basically the setting that maximizes the resolution, that is, clarity, of the image in bright field microscopy, which is what we'll be using in this lab. This makes it incrementally better than just using the microscope with a condenser at whatever height and aperture it might be, when you get it out of the cubbyhole. Our compound scopes actually employ a three-lens system. Two of the lenses, the ocular and the objective lenses, actually magnify, causing whatever it is that you have under the objective to appear bigger. The total magnification, or TM, of the microscope is the magnification of the ocular lenses, which is 10x, times the magnification of whichever objective you have in place. This could be the red or scanning objective, which is 4x, and this would give a total magnification of 40x. The yellow or low power objective, which is 10x, giving a TM of 100x, or it could be the high dry or high power objective, which is 40x. There's also a white or oil immersion objective, which is 100x, but we're not going to be using this because it requires that the space between the objective and whatever it is you're looking at be filled with oil. Without the oil, this objective is pretty much useless. Now, the third lens in our three-lens system is the one under the stage, the condenser, which focuses light from the light source into a tiny intense spotlight to illuminate the thing that you're looking at under the objective lens. When you set your scope to ocular illumination, it's this condenser that you're setting to optimize the resolving power of your scope. You'll start by recording some observations about the letter E slide, and after that, you'll be using the microscope to record observations of plant cells and animal cells. I know the handout makes it seem like you're putting the scopes away, but don't put your scopes away until you're done with them. For the plant cell observation, you'll be using the thin, paper-like membrane between the crisp layers of an onion. Don't try to smash an onion chunk. This won't work. You'll be making a wet mount, which means you place your sample, the tiny piece of fresh onion skin, flat on a glass slide and covering it with a drop of saffron and stain, then putting a cover slip on top. Make sure that you don't have stain or any kind of moisture on the bottom or sides of your slide because they'll be in contact with the microscope stage. Now from the onion membrane slide, you should be able to document all the items listed in the lab handout, as well as some of the membranous structures within the cells, like endoplasmic reticula in the area near the nucleus. For the animal cell observation, you'll be using some cells that you scrape off the inside of your cheek with the flat of a toothpick. Follow the instructions in the handout. These cells will be a whole lot less interesting than the plant cells, except hey, they are your cells. Now before you put your scope away, make sure that there's no stain or water on the stage, on the objective lenses, or any goopy or dusty stuff that fell off of your face and onto the ocular lenses. You can use a chem wipe moistened with methanol to clean the surfaces of the scope before putting it away using the method described in the handout. I'll see you guys in lab.